Assalamualaikum and hi everyone. We are from Group 8. We'll be presenting about antimicrobial activity of betel leaf extractions against microorganisms. So here is our group members. Firstly, myself, Muhammad Nasrullah bin Ibrahim. One is FARHAN bin SAMSUDIN, Muhammad RIJALUDDin bin OMAR, and lastly is HUZAIFAH Anas bin ZAKARIA. Before proceeding with our presentation today, students should be able to firstly describe betel leaf and its traditional use, explain the phytochemical compound present in betel leaf, discuss the antimicrobial activity of betel leaf against bacteria, fungi, and virus. Thirdly, explain the role of betel leaf in disease prevention, and lastly, integrate scientific evidence with Islamic perspectives. First, let's us understand what betel leaf is. Betel leaf, scientifically known as Paper betel L, is a medicinal plant that belongs to the Papyraceae family. It is commonly used as down cereal and it's widely cultivated throughout Malaysia and other Southeast Asia countries. For centuries, betel leaf has been used in traditional medicine due to its various therapeutic properties. Traditionally, it has been used to maintain the oral hygiene and freshening the breath, promote wound healing, treat bacterial and fungal infections, and relieve cough and inflammations. So these medicinal benefits are mainly attributed to several bioactivity compounds found in betel leaf. Among the most important compounds are hydroxyclavicol, eugenol, clavicol, and flavonoids. So these compounds are known to possess antimicrobial properties that help inhibit the growth of harmful microorganisms. To obtain this bioactive compound, the betel leaf must first undergo an extraction process. So the purpose of the extraction is to obtain and concentrate the compound responsible for its antimicrobial activity. So common solvents used for extractions include water, ethanol, and methanol. So among these, methanol is the most commonly used solvent because it is effective in extracting a wide range of bioactive compounds. Next, we proceed to extraction procedure of the betel leaf. First, fresh betel leaf are collected and washed to remove dirt and contaminate. The leaves are then dried to reduce moisture content and ground into the fine powder. After that, the powder is extracted using the suitable solvents such as ethanol. The extract is then filtered to remove plant residues and finally concentrated to obtain the bioactive compounds responsible for its antimicrobial activity. The concentrated extract is later used for antimicrobial testing against the microorganisms.
Next, we are going to look at the phytochemistry of the betel leaf. Betel leaf contains these compounds which are responsible for the medical and its antimicrobial properties, which is hydroxyclavicol, clavicol, eugenol, tannins, and flavonoids. First, hydroxyclavicol, which is considered the major bioactive compound in betel leaf and is largely responsible for its antimicrobial properties. It works by disrupting the bacterial cell membrane, causing leakage of important cellular content such as ions and nutrients. It can also induce oxidative stress within the microbial cell and inhibit biofilm formation. As a result, bacteria will lose their structural integrity and eventually will die. Next is clavicol, another phenolic compound found in betel leaf. Clavicol damages the bacterial cell walls and membranes, making it difficult for bacteria to maintain normal cellular functions. It also interferes with enzymes involved in bacterial metabolism, which inhibits bacterial growth and reproduction. Next compound is eugenol, which is also found in olive oil and is widely used in dentistry. Eugenol exerts its antimicrobial effect by disrupting the bacterial membrane and denaturing proteins and enzymes that are essential for bacterial survival. In addition, it inhibits ATP production, reducing the energy supply needed for bacterial growth. This helps prevent bacterial colonization and infection. Next, betel leaf also contains flavonoids. Flavonoids possess antimicrobial and antioxidant properties, which is they inhibit bacterial growth by interfering with nucleic acid synthesis and bacterial enzyme required for replication. Their antioxidant activity also helps protect host tissue from oxidative damage during infection. And lastly, tannins contribute to the antimicrobial activity of betel leaf by binding to microbial proteins and enzymes. This process causes protein precipitation and disrupt normal bacterial function. Tannins also inhibit bacterial adhesion to surfaces, preventing colonization and biofilm formation.
Now, I will explain about the antibacterial activity of betel leaf extract. Betel leaf contains several bioactive compounds such as hydroxycavicol, cavicol, eugenol, flavonoid, and tannins. These compounds are responsible for the antibacterial properties. The antibacterial mechanism works in several ways. First, betel leaf extract can damage the bacterial cell membrane. When the membrane is damaged, the essential substance inside the bacteria will leak out, causing the bacteria to die. Second, it can inhibit bacterial growth, means the bacteria cannot multiply effectively. As a result, the spread of infection can be reduced. Third, betel leaf extract can disrupt bacterial metabolism, interfering the important cellular processes needed for bacterial survival. Several bacteria has been shown to be sensitive to betel leaf extract. This includes Streptococcus mutans, Staphylococcus aureus, E. coli, and Pseudomonas aeruginosa. This antifungal effect is especially significant in the dentistry because oral fungal infections are commonly seen in denture wearers, immunocompromised patients, and individuals with poor oral hygiene. As a result, betel leaf extract has potential application in natural antimicrobial products such as mouthwashes, oral rinse, and other oral healthcare products. Besides the antibacterial activity, betel leaf also exhibit antifungal activity. One of the most important fungi affected by betel leaf is Candida albicans. This fungus is commonly found in the oral. This fungus can cause an infection known as oral thrush or oral candidiasis when it grows excessively. The antifungal mechanism is quite similar to its antibacterial action. The active compound in betel leaf can damage the fungus cell membrane, causing the structural damage to the fungus cells. In addition, betel leaf extract also can inhibit the fungal growth and reproduction, which means it will prevent the fungus from spreading and forming a more larger. Besides the antibacterial activity, betel leaf also exhibit antifungal activity. One of the most important fungi affected by betel leaf is Candida albicans. This fungus is commonly found in the oral. This fungus can cause an infection known as oral thrush or oral candidiasis when it grows excessively. The antifungal mechanism is quite similar to its antibacterial action. The active compound in betel leaf can damage the fungus cell membrane, causing the structural damage to the fungus cells. In addition, betel leaf extract also can inhibit the fungal growth and reproduction, which means it will prevent the fungus from spreading and forming more larger colonies.
Okay, now we will continue with prevention of disease. Betel leaf extract help prevent disease by inhibiting the growth of harmful microorganisms such as prevent dental caries by reducing Streptococcus mutant, prevent oral candidiasis by inhibiting Candida albicans, and reduce risk of wound infection caused by Staphylococcus aureus. Okay, now we'll continue with application in healthcare. We have 3 applications. The first one is oral healthcare, which is contain mouthwash, formulation, plaque control, prevention of dental caries, and reduction of halitosis, which is bad breath. Second, we have medical application, which is natural antiseptic agent, wound care product, and antimicrobial gel and cream. And third, we have pharmaceutical research, which is contain herbal antimicrobial product and potential alternative to synthetic antimicrobial. Betel leaf also have advantages and limitation. Advantages is natural and widely available, cost effective, contain multiple bioactive product, and potentially lower the risk of bacterial resistance. Limitation is lack of standardized dosage, antimicrobial effect varies with extraction method, less potent, and more clinical studies needed. Now, we will continue with Islamic perspective, which is akli and nakli. For nakli, and do not throw yourself with your own hand into destruction. Surah Al-Baqarah. Eat and drink but not be excessive. Surah Al-A'raf. The interpretation is Islam encourage disease prevention and natural resources should be utilized responsibility for human benefit. For akli, betel leaf contains scientifically proven antimicrobial compound. It supports the health preservation, which is hifzun-nas. It's also aligned with objective of maqasid syariah. Alhamdulillah. Okay, lastly, for conclusion, betel leaf, which is Piper betel, has effective antimicrobial properties against various bacteria and fungi. Its bioactive compound help prevent infection and support oral and general health. The use of betel leaf is supported by both scientific evidence and Islamic principle of preserving health. Further research is needed to standardize its clinical application. That's all from us. Thank you.